Hello, and welcome back to the Sleepy Darwin. Whether you're here to let your thoughts dissolve into dreams, to unwind from the day's tangles, or to simply drift through the quiet corners of nature, you've found exactly the right place. Tonight, we're venturing into a world that exists mostly out of sight, threading through soil and wood, connecting trees in conversations we're only just beginning to understand. They've been here far longer than we have, quietly building, breaking down, and rebuilding the world beneath our feet. This is the story of fungi, the silent architects of our planet, whose networks stretch further than our cities, and whose patience is measured in centuries. Now, before we descend into this underground kingdom, I have a small favor to ask. If you enjoy these gentle explorations into nature's mysteries, a like and a subscribe would mean the world to this little corner of calm content. And please, let me know in the comments where you're listening from and what time it is where you are. Are you tucked away in a forest cabin, or perhaps in a bustling apartment where mushrooms only appear on pizza? As always, I hope this video isn't too fascinating. But if it is, and you find yourself wide awake contemplating the wood wide web and mycorrhizal networks, at least you'll understand why that fallen log in the forest is actually a bustling city of decomposition and renewal. So, settle in, breathe slowly, and let's follow the mycelium into the dark. Let's begin our journey by traveling backward through time. Not just a few centuries or even millennia, but deep, deep into Earth's story back 1.5 billion years, to a world you wouldn't recognize. Close your eyes if you'd like, and imagine, before trees ever dreamed of touching sky, before flowers had even considered the possibility of petals, before the first dinosaur egg was laid, before the first fin became a foot, before any creature had drawn breath into lungs that didn't yet exist, back to a time when Earth was, well, rather lonely actually. The land was bare rock, sculpted by wind and rain into endless lifeless landscapes. The oceans teemed with life, microscopic mostly. Some early experiments in multicellularity, but the continents, they were essentially barren. Sterile stone stretching from horizon to horizon, baking under a sun that seemed to shine on nothing but mineral and dust. But somewhere, perhaps in the damp shadows where land met sea, or in the thin films of water that collected in rock crevices after rain, something remarkable was happening. Life was preparing to make one of its boldest moves, yet the colonization of land. And the pioneers of this impossible venture, they weren't plants, as you might expect. They were fungi, or rather, the ancient ancestors of what we now call fungi, organisms that had already discovered something revolutionary: how to eat rock. You see, fungi had developed something rather special: enzymes. Think of enzymes as molecular keys, each one designed to unlock specific chemical bonds. While other early life forms were content to drift in the ocean, absorbing dissolved nutrients, these proto-fungi were secreting powerful chemicals that could break down minerals. They were, in essence, the first chemists, running experiments in dissolution and transformation long before the periodic table was even a glimmer in the universe's eye. These ancient fungi, we're talking about organisms from around 1.5 to 1 billion years ago, were doing something that seems almost impossibly patient to our modern sensibilities. They were turning stone into soil. Not quickly, mind you. This wasn't some dramatic transformation you could watch happening. This was slow work, the kind measured in centuries and millennia. Imagine watching a mountain erode one grain at a time. Except instead of wind or water doing the work, it was life itself, quietly, persistently, dissolving the seemingly eternal. The fungi would extend their thread-like hyphae microscopic tubular structures thinner than spider's silk into the tiniest cracks in rocks. There, they'd release their cocktail of acids and enzymes. Slowly, minerals would dissolve. Nutrients locked away in stone for millions of years would suddenly become available. And bit by bit, grain by grain, the fungi were creating something Earth had never seen before: soil. That dark, rich, life-giving substance we so take for granted today, every handful of it owes its existence to these ancient fungal pioneers. But here's where the story becomes even more extraordinary. Around 600 to 400 million years ago, as the Ordovician period gave way to the Silurian and then the Devonian, plants were beginning their own tentative exploration of land. These weren't the mighty oaks or graceful ferns we know today. These were simple things, perhaps similar to modern liverworts or mosses, low, humble, clinging to wet places, and they faced a terrible problem. They had no roots. Can you imagine trying to make a living on bare rock with no way to anchor yourself? no way to reach nutrients locked in stone. It would be like trying to build a house with no foundation and no tools. The early plants had photosynthesis. They could make sugar from sunlight, water, and carbon dioxide. But they needed minerals, nutrients, stability. They needed what the fungi had. And so, somewhere in those ancient days, a partnership was formed. A handshake deal, as it were, that has lasted over 400 million years and shows no signs of ending. The fungi would provide minerals, water, and anchorage. The plants would provide sugars, those sweet products of photosynthesis that fungi, lacking chlorophyll, couldn't make themselves. It was perhaps the most important business partnership in Earth's history. Even today, over 90% of all plant species maintain this ancient contract with fungi. Those mycorrhizal relationships from the Greek words for fungus and root are still the foundation of almost every ecosystem on land. Think about that for a moment. Every forest you've ever walked through, every meadow that's ever made you want to lie down in the grass, every flower that's ever caught your eye, all of it exists because of a deal made before bones existed, before eyes evolved to see colors, before anything had ever thought a thought. It's rather humbling, isn't it? And this partnership didn't just allow plants to survive on land, it transformed the entire planet. You see, as plants spread across the continents, powered by their fungal partners, they began to change the atmosphere itself. They pulled carbon dioxide from the air in quantities that had never been seen before. They released oxygen as a waste product of photosynthesis. The fungi, meanwhile, were breaking down rock at an accelerating pace, releasing minerals that would run off into the oceans, changing ocean chemistry, affecting global climate patterns. Every breath you take has fungi to thank, in a roundabout, ancient sort of way. The oxygen levels that allow us mammals to exist, they're here because fungi helped plants colonize land, the soil that grows our food. Fungi created the prototype, the stable climate that allowed civilization to develop. Fungi helped regulate it by assisting in the carbon cycle. We live on a fungal planet, really, even if we rarely notice our silent partners. Now, speaking of things we rarely notice, let me tell you about one of paleontology's most enduring mysteries. A puzzle that takes us to the Devonian period, roughly 420 to 370 million years ago. Picture, if you will, a landscape unlike anything on modern Earth. The first forests are just beginning to form, but they're not quite forests as we know them. The tallest trees might reach 30 feet. Many are more like overgrown ferns or club mosses. The ground is covered with primitive plants, and the air buzzes with early insects. But rising above it all, scattered across this ancient landscape like monuments from an alien civilization, stand massive spires. They're up to 26 feet tall and 3 feet wide. Imagine a telephone pole made entirely of living tissue. For over a century, these structures, known from fossils as prototaxites, have puzzled scientists. Were they enormous fungi? Lichens? Some kind of plant we no longer have any modern equivalent for? The evidence increasingly points to fungi. The internal structure, when examined under microscopes, shows what appear to be fungal hyphae. The chemical signatures match what we'd expect from fungi. But still, the debate continues, because the implications are so extraordinary. Imagine a world where the tallest things weren't trees, but mushrooms reaching toward prehistoric skies. Where you might walk through a landscape dominated not by wooden trunks but by fungal towers, each one possibly taking centuries to grow, each one a slow-motion monument to patience. What would it have been like, do you think, to stand in the shadow of a 26-foot mushroom? The light would have been different, filtered not through leaves but perhaps through nothing at all, as these giants might have been solid columns rather than umbrella-shaped like modern mushrooms. The smell would have been different too, that earthy fungal scent we associate with autumn forests, but magnified, ancient, unmarked by any of the familiar fragrances we know from flowering plants, because flowers hadn't evolved yet. These prototaxites, whatever they were exactly, dominated landscapes for at least 50 million years. That's far longer than grass has existed, longer than flowers have been colouring the world, longer than mammals have been nursing their young. And then, as mysteriously as they appeared in the fossil record, they vanished. Perhaps they were outcompeted by the rising forests. Perhaps climate change, and yes, Earth has seen many climate changes long before humans arrived, made their particular way of life impossible. We may never know for certain. But what strikes me most about prototaxites isn't just their size or their mystery. It's what they represent about the fungal approach to existence. These organisms, if they were fungi, weren't trying to dominate their environment the way we think of dominance. They weren't spreading leaves to catch all the sunlight or growing thorns to fight off competitors. They were just being, growing slowly, processing nutrients, probably providing habitat for countless smaller organisms, and reshaping their world not through force but through persistence. And this brings us to something I often wonder about when I'm thinking about fungi late at night. Perhaps when I can't sleep and my mind wanders to these ancient architects. What does it mean to be successful in evolutionary terms? We humans often think of success as dominance being the strongest, the fastest, the smartest. We build civilizations that reach toward the sky, much like those ancient prototaxites, and we measure our worth in how much we can control, how much we can change. But fungi offer a completely different model of success. Their strategy has never been domination, it's been integration. Instead of standing apart from other organisms, they weave themselves into the fabric of life so thoroughly that removing them would cause everything else to collapse. They don't compete so much as they facilitate. They don't conquer territories. They connect them. Consider this. Fungi have survived at least 5 major mass extinctions. They've watched the rise and fall of the dinosaurs, the emergence and disappearance of countless species. They were here before the Atlantic Ocean opened, and they'll probably be here long after it closes again in the deep future. While empires rise and fall, while species explode into diversity and then vanish into fossil beds, fungi just keep threading through it all. Quiet, patient, essential. There's something deeply comforting in that, I think. In our human lives, we often feel pressure to make our mark, to achieve something lasting, to build monuments to our existence. But fungi suggest another way of being significant, not through monuments but through relationships, not through dominance but through indispensability. They remind us that sometimes the most profound influence is the one that goes unnoticed, the one that works so quietly and consistently that it becomes part of the foundation itself. When we look at human history, we celebrate the builders of pyramids, the conquerors of nations, the inventors of revolutionary technologies. But what if we celebrated the network builders instead? The ones who connected communities, who facilitated trade and communication, who broke down barriers and recycled resources. What if success wasn't about standing tallest, but about supporting the most life? The fungi seem to have figured this out more than 1 billion years ago. While other organisms were learning to compete, fungi were learning to collaborate. While others were building walls, fungi were building bridges, microscopic bridges, yes. But bridges that would eventually connect entire forests in networks of mutual support. And that's what we're going to explore next, those incredible networks that fungi have built beneath our feet. Networks so vast and complex that scientists have only recently begun to understand them. Networks that some researchers now call the Wood Wide Web, though that playful name hardly does justice to the profound implications of what's happening down there in the darkness. But for now, just rest with this thought. Beneath you, right now, wherever you're listening to this, there are fungal threads. They've been there long before you arrived, and they'll be there long after you leave. They're not concerned with the brief flicker of human time. They're operating on geological timescales, with a patience that borders on the eternal. They're the ancient builders, still at work, still weaving the world together, one hypha at a time. The story of fungi is really the story of time itself. Deep time, patient time, the kind of time that transforms continents and creates atmospheres. It's a story that makes our human urgencies seem both precious and fleeting, like mushrooms that appear overnight after rain, beautiful, temporary, but part of something much, much larger. In the 1990s, a young forest ecologist named Suzanne Simard was doing something that made her colleagues rather skeptical. She was injecting trees with radioactive carbon dioxide, not to harm them, you understand, but to trace where the carbon went. She had a hypothesis that seemed, at the time, almost like fantasy. She believed trees were sharing resources underground, connected by fungal networks in ways that would fundamentally change how we understand forests. The scientific establishment was, let's say, resistant. The idea that trees might be cooperating rather than simply competing went against decades of forestry doctrine. Competition for light, water, and nutrients was supposed to be the driving force of forest dynamics. The notion that trees might actually be helping each other? That sounded more like a children's story than serious science. Trees talking? It sounds like a fairy tale, but the science, as Simard would prove, is even more magical. What Simard found with her radioactive tracers was revolutionary. The carbon she injected into Douglas fir trees didn't stay in the Douglas firs. It moved. It traveled through the soil, through fungal networks, and appeared in nearby paper birch trees. And not just randomly, the transfer increased when the birch trees were shaded and decreased when they had plenty of light. The trees were actually sharing resources based on need. But here's where it gets even more extraordinary. When Simard reversed the experiment, injecting the birch trees with tracers, she found carbon moving back to the Douglas firs, particularly to the smaller, shaded firs that needed it most. This wasn't just one-way charity. This was reciprocal exchange. A forest economy running on principles we're only beginning to understand. The moment scientists realized forests were superorganisms rather than collections of individual trees competing for survival, was like the moment astronomy realized the Earth wasn't the center of the universe. Everything had to be reconsidered. Those solitary oaks standing in meadows? Not solitary at all. That grove of aspens that all turn golden at exactly the same moment in autumn? They might share roots, but they're also connected by something else. Something that weaves between and around their roots like neural pathways in a vast, dispersed brain. The forest floor, it turns out, is absolutely riddled with fungal threads, millions of miles of them in a single handful of forest soil. These mycorrhizal networks, from the Greek words for fungus and root, come in two main varieties. And understanding the difference helps us appreciate just how intimate these partnerships are. Ectomycorrhizal fungi wrap around tree roots like a soft glove, never quite penetrating the root cells but forming an incredibly intimate embrace. If you've ever seen those prized truffle hunting pigs in France, they're searching for the fruiting bodies of ectomycorrhizal fungi that partner with oak and hazel trees. These fungi tend to partner with trees in temperate and boreal forests, your pines, firs, oaks, and birches. Endomycorrhizal fungi, on the other hand, are even more intimate. They actually penetrate root cells, branching inside them to form tiny tree-like structures called arbuscules. It's like having a house guest who doesn't just stay in your spare room but actually sets up shop inside your living room walls. These fungi partner with about 85% of all plant species, from grasses to tropical trees to the tomatoes in your garden. The mechanics of these root fungus partnerships are beautifully simple and breathtakingly complex at the same time. The basic transaction is straightforward. Carbon for nutrients. The trees, through photosynthesis, create sugars anywhere from 20 to 80% of which they might send down to their fungal partners. In return, the fungi provide minerals, particularly phosphorus and nitrogen, but also water and trace elements the trees couldn't access on their own. Think of it as nature's internet, but instead of cat videos, they're sharing nutrients, and like our internet, the bandwidth varies. Some connections are fiber optic highways, others are more like dial-up. A large tree might be connected to hundreds of other trees through dozens of fungal species, creating a network complexity that makes our World Wide Web look almost simple by comparison. The currency of this forest economy is surprisingly sophisticated. It's not just sugar for minerals like some simple barter system. The trees can actually adjust their payments based on market conditions. When phosphorus is scarce in the soil, trees pay more sugar for it. When sugar is scarce, maybe during a cloudy summer, the exchange rate shifts. There's even evidence of trees maintaining relationships with multiple fungal partners simultaneously, perhaps to ensure better rates or more reliable service. And the fungi? They're not passive conduits. They're active traders, sometimes holding resources when prices are low and releasing them when they can get better returns. They can even facilitate trades between trees of different species, taking their cut as commission. A fungus might accept summer sugars from an oak tree and provide minerals in return, then trade those minerals to a pine tree for winter sugars when the deciduous oak is dormant. It's an economy that never closes, a market that's been running for hundreds of millions of years. But perhaps the most profound discovery about these networks involves what Simard calls mother trees, the matriarchs of the forest. These are the ancient giants, the ones that have survived centuries of storms, droughts, and changes. They're not just the biggest trees in the forest, they're the most connected. A single mother tree might be linked to hundreds of younger trees through the mycorrhizal network. These hub trees play a role that's only recently become clear to science, but that indigenous peoples have long understood intuitively. They nurture their offspring, and yes, trees can recognize their own seedlings through chemical signatures in the network. Mother trees will funnel more resources to their own young than to strangers, though they'll support those strangers too if resources allow. They'll even reduce their own root competition to make room for their offspring to establish themselves. When a mother tree is dying, whether from old age, disease, or logging, something remarkable happens. In what researchers have documented as a kind of forest inheritance system, the dying tree dumps its resources into the network: carbon, nitrogen, phosphorus, chemical defense compounds. All of it flows out through the fungal networks to the surrounding trees. It's a kind of forest inheritance system, written not in wills but in root tips and fungal threads. Simard has described watching a massive Douglas fir that was dying from beetle infestation. Over the course of its decline, it transferred its carbon reserves to young trees in its network, particularly to its own offspring. Even defense chemicals that might help the younger trees resist the very beetles that were killing the mother moved through the network. The dying tree was ensuring the forest would continue, that its death would feed new life. This inheritance isn't random or equally distributed. The resources tend to flow to the trees that need them most: the shaded understory trees struggling for light, the young seedlings just getting established, the trees fighting off disease or insect attack. It's as if the forest has a built-in social safety net, a welfare system that ensures the most vulnerable get support when they need it most. But the network doesn't just move nutrients, it moves information. When a tree is attacked by insects, it doesn't just defend itself, it posts a warning to the whole neighborhood. The attacked tree releases chemical signals through its roots into the fungal network. These signals travel to connected trees, which then begin producing defense chemicals before they're even attacked. They're essentially vaccinating themselves based on intelligence from the network. The speed of these warnings can be remarkable. In some studies, trees dozens of meters away from an attacked tree began mounting chemical defenses within hours. The signals can be specific: 2 different chemical messages for different threats. A warning about aphids triggers different responses than a warning about caterpillars or fungal pathogens. Trees can even eavesdrop on each other's stress signals. A tree suffering from drought stress releases particular chemical signatures into the network. Neighboring trees detect these signals and begin closing their stomata, the tiny pores they use for gas exchange to conserve water before they themselves experience water shortage. They're learning from their neighbors' experiences, adapting based on communal knowledge rather than individual trial and error. The chemical vocabulary of these communications is astoundingly rich. Trees and fungi exchange not just simple nutrients but complex molecules: phytohormones that regulate growth, allelopathic compounds that suppress competitors, antibiotics that fight pathogens, and even what some researchers provocatively call infochemicals, molecules that carry pure information rather than nutritional value. But let's not paint too rosy a picture here. This underground network, miraculous as it is, isn't some kind of socialist utopia where all trees share equally and everyone gets along. The forest has its share of cheaters, hackers, and thieves. Even in this underground wood wide web, there are freeloaders and pirates. Some plants have evolved to tap into the mycorrhizal network without contributing anything back. These mycoheterotrophs, plants that have given up photosynthesis, entirely hack into the network and steal sugars that other plants have worked to produce. The ghostly white Indian pipe plant you might find in deep forest shade? It's a complete parasite on the network, taking carbon from trees via shared fungal partners and giving nothing in return. There are even plants that use the network for chemical warfare. Black walnut trees, for instance, release juglone, a toxic compound, into the soil around their roots. This chemical moves through the mycorrhizal network, poisoning potential competitors. The walnut trees have essentially weaponized the network, using it to clear space for themselves and their offspring. And the fungi themselves aren't always honest brokers. Some species are better at extracting payment than providing services. They might hoard resources when trees need them most, driving up the price of their services. Others might preferentially direct resources to trees that pay better, regardless of need. The network can amplify inequalities as easily as it can reduce them. Competition and cooperation exist simultaneously in these networks, often between the same organisms. 2 trees might share resources through fungal networks while simultaneously competing for light above ground. A fungus might nurture a tree seedling one season and parasitize it the next if conditions change. The boundaries between mutualism and exploitation are constantly shifting, negotiated moment by moment through chemical signals we're only beginning to decode. This darker side of the network shouldn't surprise us, really. No system in nature is purely cooperative or purely competitive. Life is always finding balance between self-interest and mutual benefit, between competition and collaboration. The mycorrhizal network is powerful precisely because it allows for both. creating a flexible system that can adapt to changing conditions. What strikes me most profoundly about these fungal networks is how they challenge our human notions of individuality. We're so used to thinking of ourselves as separate beings, distinct and independent. But trees in a forest aren't individuals in any meaningful sense. They're nodes in a network, each one inseparable from the whole. A tree without its fungal partners is like a brain without a body. It might survive briefly, but it can't truly function. Perhaps loneliness is just forgetting we're all connected, even when we can't see the threads. We humans, too, are never as isolated as we imagine. We're connected through language, culture, shared air and water, the microbes we exchange, the foods we eat that come from soils built by fungi. We're part of networks we rarely perceive, influenced by connections we don't consciously recognize. What could human networks learn from fungal ones? The mycorrhizal web suggests that resilience comes from redundancy, multiple connections, multiple pathways, no single point of failure. It shows that diversity strengthens the whole system. Forests with more tree species and more fungal species are more resistant to disease, drought, and disturbance. It demonstrates that supporting the vulnerable members of a network ultimately benefits everyone, creating a more stable and productive system overall. The network also embodies a kind of patience we humans rarely achieve. These fungal networks develop over decades, centuries, millennia. They're not built for quick profits or short-term gains. They're built for persistence, for continuity, for the long game of survival. A fungal network might take 50 years to fully connect a forest, but once established, it can persist for thousands of years. In our age of social media and instant global communication, we might think we've invented connectivity, but forests have been doing social networking for hundreds of millions of years, and their version might be more sophisticated than ours. Their network doesn't spread misinformation or amplify outrage. It shares resources based on need, communicates warnings that save lives, and maintains connections that can last for centuries. There's something both humbling and hopeful in learning about these networks. Humbling because it reminds us that intelligence and communication aren't unique to humans or even to animals. The forest is thinking, in its own way, processing information, making decisions, solving problems. And hopeful because it suggests that cooperation and mutual aid aren't human inventions, but fundamental patterns of life, written into the very foundations of ecosystems. The Wood Wide Web reminds us that the trees we see are just the visible portion of something much larger and more complex. Like icebergs, most of their story is hidden beneath the surface, in the dark, humid world of soil and root and fungal thread. When we walk through a forest, we're walking above a network of communication and exchange that makes our internet look primitive by comparison. We're walking above conversations that have been going on for centuries, above relationships older than our oldest cities. Every footstep in a forest is a footstep on history, on community, on an economy we're only beginning to understand. The mushrooms we see are just the fruiting bodies of this vast network, temporary structures compared to the permanent infrastructure below. The forest floor beneath our feet is alive, with messages we can't hear, with exchanges we can't see, with connections that bind the forest into something greater than the sum of its parts. And perhaps that's the most important lesson of the Wood Wide Web, that cooperation is as fundamental to life as competition, that sharing resources can be a successful survival strategy, that networks of mutual support are not human inventions, but ancient patterns that have shaped life on Earth for hundreds of millions of years. In a time when human society often feels fractured and disconnected, the fungal networks beneath our feet offer a different model, one of connection, communication, and community that has stood the test of deep time. There's a thought experiment I'd like you to consider. Imagine if nothing ever broke down, if every leaf that fell, every tree that died, every creature that breathed its last breath simply remained intact forever. Within a few thousand years, a blink in geological time, we'd be buried in the bodies of everything that ever lived. The earth would be a monument to death, layers upon layers of preserved remains, with no room for new life to emerge. But this doesn't happen, and we have fungi to thank for that. They are nature's ultimate recyclers, the patient dismantlers of everything life builds. While plants are busy constructing, turning air and water and sunlight into wood and leaf and flower, fungi are quietly deconstructing, breaking these elaborate structures back down into their component parts, making them available for life to use again. Without decomposition, life as we know it would be impossible. All the carbon would eventually be locked up in dead bodies. All the nitrogen, all the phosphorus, all the essential elements of life would be frozen in place, unavailable. The great cycles that keep our planet habitable, the carbon cycle, the nitrogen cycle, the water cycle, would grind to a halt. Earth would become a beautiful mausoleum, perfectly preserved and utterly dead. But fungi won't let that happen. They're constantly at work, breaking apart what life puts together, with a patience that borders on the eternal. A fallen tree might take decades to fully decompose, but fungi will be there for every moment of it, slowly, methodically, returning the tree to the soil from which it grew. The chemistry of decay is more sophisticated than anything happening in our most advanced laboratories. Fungi produce an astounding array of enzymes, biological catalysts that speed up chemical reactions. Think of enzymes as molecular scissors, each type designed to cut specific chemical bonds. Some snip proteins into amino acids, others cleave starches into simple sugars, still others break down fats into fatty acids and glycerol. But the real magic happens with lignin, the compound that makes wood woody. Lignin is one of the most complex organic polymers on Earth, a three-dimensional maze of aromatic rings and carbon bonds that gives trees their strength and rigidity. It's so complex that most organisms can't break it down at all. Bacteria, for all their metabolic diversity, mostly can't handle lignin, neither can animals. If you eat wood, it passes through you essentially unchanged. Only fungi, specifically white rot and brown rot fungi, have truly mastered the chemistry of lignin degradation. They produce specialized enzymes like lignin peroxidase, manganese peroxidase, and laccases that can actually break apart lignin's formidable structure. These enzymes generate highly reactive molecules, free radicals that attack lignin almost randomly, breaking it apart piece by piece like a molecular wrecking ball. They're running a chemistry lab that would make any scientist envious, all without a single beaker, without temperature controls, without any of the equipment we consider essential for complex chemistry. They're doing it in the wild, in the rain and snow, in the heat of summer and the cold of winter, with a reliability that our best industrial processes can't match. The process is so sophisticated that we still don't fully understand it. We've identified many of the enzymes involved. We can track some of the intermediate compounds, but the complete pathway from intact lignin to fully degraded components remains partially mysterious. Nature's chemistry is humbling like that. What fungi do, casually, automatically, we struggle to replicate even with our best technology. White rot fungi attack both lignin and cellulose, leaving wood bleached and stringy. If you've ever seen a log that looks almost white, with a texture like shredded paper, you've seen their work. Brown rot fungi, on the other hand, preferentially attack cellulose while leaving much of the lignin behind, creating that crumbly, brown, cube-like rot you might find in old timber. The decomposition of a fallen tree follows a predictable succession, a careful choreography of fungal arrivals and departures. It begins even before the tree falls. Endophytic fungi species that live within living trees without causing disease are already present in the wood. When the tree dies, these fungi are first to begin the decomposition process, having a head start on all the others. Next come the primary colonizers, the fungi that fruit within weeks or months of the tree's death. These are often species like oyster mushrooms or turkey tail fungi, aggressive decomposers that quickly claim territory on the newly available resource. They attack the easily digestible compounds, the sugars, starches, and simple proteins that are still present in the recently dead wood. As these easy pickings are consumed, secondary decomposers arrive. These species are adapted to break down more complex compounds. They might specialize in particular types of wood or prefer certain moisture conditions. Some attack from the outside in, others from the inside out. Some follow beetle galleries, using the paths carved by insects as highways into the wood's interior. Finally come the tertiary decomposers, the cleanup crew that handles the most recalcitrant compounds. By this stage, the log might barely resemble wood anymore. It's soft, crumbly, maybe held together more by moss and other plants growing on it than by its own structure. These late-stage fungi are breaking down the last of the lignin, the final complex polymers, returning the last of the tree's carbon to the atmosphere and soil. It's like a very slow, very important relay race where everyone wins. Each group of fungi prepares the way for the next, breaking down certain compounds and leaving others, creating conditions that favor different species at different times. The primary colonizers couldn't survive in the nutrient-poor environment of late-stage decomposition, and the tertiary decomposers couldn't compete with the aggressive primary species on fresh wood. This succession can take decades for a large tree. In tropical rainforests, where decomposition is rapid, a fallen tree might disappear in 5 to 10 years. In boreal forests, where cold temperatures slow fungal metabolism, the same process might take a century or more. In particularly dry or cold environments, wood can persist for thousands of years, decomposing so slowly that it seems almost eternal from a human perspective. As fungi decompose organic matter, they create something precious: humus. This dark, complex material is the foundation of soil fertility. Humus isn't just decomposed organic matter, it's organic matter that has been processed, transformed, complexified by fungal and bacterial action into something new. It's a stable, carbon-rich substance that can persist in soil for hundreds or even thousands of years. The transformation from log to earth is a kind of alchemy. The fungi take the ordered structure of wood, its careful arrangement of cellulose, lignin, and other compounds, and break it down into chaos. But from that chaos emerges a new kind of order: the complex chemistry of humus, with its ability to hold nutrients, retain water, and support new life. Humus is chemically complex beyond our full understanding. It contains thousands of different compounds, many of which we haven't even identified. It has properties that seem almost magical. It can hold many times its weight in water. It can bind and release nutrients as plants need them. It can buffer pH changes. It can even bind and neutralize certain toxins. Every handful of forest soil is a graveyard and a nursery all at once. It contains the transformed remains of countless organisms: trees, leaves, insects, bacteria, fungi themselves, all processed and reprocessed until their original identity is completely lost. But it also contains the potential for new life, the nutrients and structure and moisture that seeds need to germinate, that fungal spores need to grow, that countless soil organisms need to survive. The fungi don't just create humus through decomposition, they also contribute their own bodies to it. Fungal hyphae, those microscopic threads that permeate soil, are constantly growing and dying. A significant portion of soil organic matter, perhaps 30% or more in some soils, consists of dead and living fungal biomass. The fungi are literally building soil with their own bodies, creating the matrix in which future life will grow. There's a particular beauty in decomposition that our culture often overlooks. We tend to see decay as ugly, as something to be avoided or hidden, but spend time watching fungi decompose a log, and you might change your mind. The progression of colors as different fungi colonize wood, the blacks and browns of early decomposition, the whites of lignin degradation, the oranges and yellows of various bracket fungi, is like a slow-motion sunset. Some fungi add an element of magic to decomposition: bioluminescence. In forests around the world, from the Amazon to the Pacific Northwest to the bamboo groves of Japan, certain fungi glow in the dark. The ghostly green light of foxfire, as it's sometimes called, has inspired folklore and fear for millennia. These ghost lights in the forest are actually fungi like honey mushrooms or bitter oyster mushrooms, producing light through the same chemical reaction that makes fireflies glow. The purpose of fungal bioluminescence remains debated. Perhaps it attracts insects that help disperse spores. Perhaps it's a byproduct of metabolism with no adaptive function. Or perhaps, and this is pure speculation, it serves some purpose we haven't yet imagined. Whatever the reason, there's something profound about decomposition that literally brings light to darkness. The aesthetics of fungal decomposition go beyond the visual. There's the smell of decomposition, not the putrid smell of anaerobic decay, but the rich, earthy smell of healthy decomposition. That smell, called petrichor when it rises from soil after rain, is partly produced by fungi. It's the smell of transformation, of nutrients being liberated, of the endless cycle of life and death and rebirth. There's also the texture of decomposition. Run your hand over a log covered in turkey tail fungi. Feel the velvety zones of color, the tough but flexible brackets. Press your finger into wood softened by years of fungal decay. Feel how it yields and crumbles. These textures tell the story of chemistry in action, of complex polymers being methodically disassembled. Finding peace in impermanence is perhaps one of fungi's greatest teachings. They show us that decomposition isn't an ending but a transformation. That tree that falls in the forest doesn't disappear. It becomes soil. It feeds new trees. It provides habitat for countless organisms during its slow dissolution. The carbon in its trunk might become part of a fungus. then an insect that eats the fungus, then a bird that eats the insect, then back to soil when the bird dies. Nothing is lost, only transformed. In Japanese aesthetics, there's a concept called mono no aware, the pathos of things, the gentle sadness of transience. Fungi embody this principle perfectly. Their fruiting bodies emerge overnight and disappear just as quickly. The logs they decompose slowly fade from wood to soil. Everything changes, nothing remains, and there's a deep beauty in this constant transformation. Fungi also demonstrate that decomposition is creative. As they break down dead organic matter, they create new structures, the intricate patterns of mycelial networks in rotting wood, the fantastic shapes of bracket fungi, the fairy rings that appear in grasslands. Destruction and creation are not opposites in the fungal world, but part of the same process. We live in a culture that often denies or avoids thinking about decomposition. We embalm our dead, we rake up leaves, we remove fallen trees, we try to maintain the illusion of permanence, of unchanging order. But fungi remind us that decomposition is not just natural but necessary. Without it, there can be no renewal, no recycling of nutrients, no continuation of life. The decomposer's ballet is performed every day in every forest, in every garden, in every compost pile. It's slow, patient, essential work. While we're rushing about our daily lives, fungi are quietly dismantling the dead and preparing the ground for new life. They're ensuring that the great cycles continue, that nutrients flow, that life persists. Perhaps, if we paid more attention to decomposition, we might have a healthier relationship with our own mortality. We might see death not as an ending, but as a transformation, not as a loss, but as a return. We might understand that we, too, are part of these great cycles, that our bodies will one day feed the fungi that feed the trees that produce the oxygen that allows new life to breathe. The fungi don't mourn the trees they decompose. They simply transform them, patiently, thoroughly, beautifully. They take apart what life has put together, not as an act of destruction but as an act of renewal. They ensure that every ending is also a beginning, that every death feeds new life, that nothing is ever truly lost. In this endless recycling, this patient dismantling and rebuilding, there's a profound comfort. We are not separate from these cycles but part of them. The carbon in our bodies has been through countless organisms before us and will pass through countless more after we're gone. We are temporary arrangements of ancient atoms, briefly conscious, briefly alive, but always part of the larger dance of matter and energy that fungi help choreograph. The decomposer's ballet never ends. It's been performed for over 1 billion years and will continue long after we're gone. It's a dance of patience, of chemistry, of transformation. It's the dance that makes all other dances possible, the essential recycling that keeps the planet alive. And it's happening right now, beneath your feet, in the fallen leaves outside your window, in the forgotten corners where fungi quietly, persistently, beautifully unmake the world so it can be made anew. When we think of fungi, most of us picture mushrooms, those umbrella-shaped structures that pop up after rain. But limiting our understanding of fungi to mushrooms is like thinking all transportation is cars while ignoring planes, trains, ships, bicycles, and feet. The fungal kingdom contains shapeshifters of extraordinary diversity, organisms that can transform their very structure depending on conditions, needs, and opportunities. It's like discovering that icebergs can also be clouds and rivers, that the same basic organism can take radically different forms. Let's start with yeasts, the single-celled wonders that have been humanity's partners since before we were fully human. Yeasts are fungi that have abandoned the multicellular lifestyle, choosing instead to exist as individual cells. They're the minimalists of the fungal world, having stripped away everything except the essentials: a cell wall, a nucleus, and the metabolic machinery to convert sugars into alcohol and carbon dioxide. Saccharomyces cerevisiae, baker's and brewer's yeast, might be humanity's oldest biotechnology. We've found evidence of fermented beverages from 90 years ago in China, and bread making goes back at least 14 years. These single cells, invisible to the naked eye, have shaped human civilization. Without yeast, we'd have no beer, no wine, no bread as we know it. Entire cultures have been built around what these microscopic fungi can do. But yeasts aren't just about human uses. In nature, they're everywhere on fruit surfaces, in nectar, in tree sap, in soil, even in the deep ocean. They've evolved to rapidly exploit sugar-rich environments, reproducing quickly when conditions are good. A single yeast cell can divide every 90 minutes under ideal conditions, turning one cell into millions in a matter of days. What's remarkable about yeasts is what they've given up to achieve this lifestyle. Most fungi grow as networks of hyphae, those thread-like structures that can explore and exploit large areas. Yeasts have abandoned this entirely, betting everything on being small, mobile, and fast reproducing. They can't form the vast networks of their multicellular cousins, can't create mushrooms, can't digest wood. But they don't need to. They've found their niche and dominated it completely. Molds represent a different strategy entirely. These are the fuzzy inventors, the fungi that create antibiotics, flavor cheeses, and occasionally ruin your forgotten leftovers. Molds grow as masses of branching hyphae, creating those familiar fuzzy coatings on bread, fruit, or shower curtains. Each strand of mold is a sophisticated chemical factory, producing enzymes to digest food and sometimes creating compounds that would make pharmaceutical companies envious. Penicillium is perhaps the most famous mold genus, having saved more human lives than any other organism except perhaps humans themselves. But Penicillium species don't produce penicillin to help us. They produce it to eliminate bacterial competition in their environment. The same genus that gives us life-saving antibiotics also gives us blue cheese, using Penicillium roqueforti to create those distinctive veins and flavors. Aspergillus, another common mold, shows just how versatile these fuzzy inventors can be. Different Aspergillus species are used to ferment soy sauce, make sake, produce citric acid for soft drinks, and create enzymes for everything from laundry detergent to biofuel production. But other Aspergillus species produce aflatoxins, some of the most carcinogenic compounds known. The same genus contains both beneficial partners and dangerous enemies. Mushrooms, those structures we most associate with fungi, are really just the tip of the fungal iceberg, temporary reproductive structures that represent maybe 5% of the organism's total biomass. The rest exists as a network of hyphae spreading through soil, wood, or whatever substrate the fungus calls home. A mushroom is to a fungus what an apple is to an apple tree, except the tree is underground and might cover several acres. The variety of mushroom forms defies easy categorization. There are the classic umbrella shapes, but also brackets that jut from trees like shelves, puffballs that explode in clouds of spores, bird's nest fungi with spore packets that look exactly like tiny eggs, earth stars that open like flowers, and stinkhorns that smell like rotting meat to attract flies. Each form represents a different solution to the same problem: how to disperse spores effectively. The life cycles of fungi contain mysteries that still puzzle scientists. Take spores, those microscopic propagules that fungi use to reproduce and disperse. A single mushroom can produce billions of spores. The giant puffball can release over 7 trillion. These spores are extraordinary in their resilience and traveling ability. Some spores ride the jet stream, traveling between continents like microscopic astronauts. They've been found in the stratosphere, in clouds, in Antarctic ice cores. Fungal spores can survive conditions that would kill most life forms. They endure extreme cold, radiation, desiccation, and vacuum. Some spores can remain viable for decades, possibly centuries, waiting for the right conditions to germinate. They're like escape pods from science fiction, carrying the complete genetic blueprint of their species in a package smaller than a grain of pollen. The reproductive strategies of fungi boggle the mind with their complexity. Many fungi can reproduce both sexually and asexually, options upon options depending on circumstances. When conditions are stable and good, they might reproduce asexually through spores or fragmentation, creating clones quickly and efficiently. When conditions become stressful or change rapidly, they switch to sexual reproduction, creating genetic diversity that might help some offspring survive new challenges. But fungal sex is nothing like what we're familiar with. Many fungi have multiple mating types, not just 2 like male and female, but sometimes thousands. Schizophyllum commune, a common bracket fungus, has over 23 mating types. Any individual can mate with any other individual except those of its own mating type. It's a system that maximizes genetic mixing while preventing inbreeding. Some fungi have abandoned sex entirely, reproducing only asexually for millions of years. Others require sex to complete their life cycle, going through elaborate dances of cellular fusion and nuclear migration. Still others can switch between sexual and asexual reproduction, or even between different types of asexual reproduction, depending on environmental cues. The morphological flexibility of fungi extends beyond just reproductive strategies. Many fungi are dimorphic, able to switch between completely different growth forms. These are the werewolves of the microbial world, transforming from one shape to another based on temperature, nutrients, or other triggers. Histoplasma capsulatum, which causes histoplasmosis, grows as a mold in soil but transforms into a yeast when it infects warm-blooded animals. The temperature change triggers a complete reorganization of its cellular structure. In soil at 25 degrees, it's a branching network of hyphae. In human lungs at 37 degrees Z, it becomes single, round yeast cells. Same genome, same organism, completely different form. Candida albicans, familiar to many as the cause of yeast infections, can switch between at least 6 different cell types. It can grow as round yeast cells, elongated pseudohyphae, true hyphae, thick-walled chlamydospores, and several other forms. Each form has different properties, some better at spreading, others better at resisting immune attack, still others better at adhering to surfaces. This shape-shifting ability isn't just a curiosity, it's often essential for survival. The different forms allow fungi to exploit different environments, evade immune systems, or respond to changing conditions. It's evolution's way of hedging bets, allowing a single organism to thrive in multiple niches. Speaking of thriving, let me tell you about the largest organism on Earth. In the Malheur National Forest in Oregon grows an Armillaria ostoi, a honey mushroom that covers 385 acres, about 3.7 square miles. This single organism, genetically uniform throughout, is estimated to be at least 400 years old, possibly much older. It weighs an estimated 0 tons, making blue whales look tiny by comparison. This giant exists mostly as rhizomorphs, root-like structures that spread through the forest soil and can penetrate tree roots. Above ground, you might see honey-colored mushrooms clustered around tree bases in autumn, but these are just the temporary fruiting bodies of an organism that extends underground for miles. It's slowly eating its way through the forest, killing trees and decomposing their roots, having turned an entire landscape into its body. The existence of such giants challenges our very concept of what an individual organism is. Is this Armillaria a single individual or a colony? Where does the organism end and the environment begin when its hyphae permeate thousands of tons of soil? These are questions that fungi force us to ask, challenging our animal-centric view of biology. Fungi haven't just conquered normal environments, they thrive in places that seem impossible for life. In the dry valleys of Antarctica, where temperatures rarely rise above freezing and liquid water is almost non-existent, fungi persist. They grow inside rocks, in the tiny spaces between mineral grains, surviving on moisture from rare snow and obtaining nutrients from the rock itself. In deep ocean vents, where temperatures exceed 100 degrees and pressure would crush most organisms, fungi thrive. They've been found at depths of over 40 meters, in sediments that have been buried for millions of years. Some deep-sea fungi can withstand pressures of over 500 atmospheres, equivalent to having 500 times Earth's atmospheric pressure pushing on every square inch of their cells. In the Chernobyl exclusion zone, fungi not only survive but thrive on radiation. Some species actually grow toward radioactive sources, using melanin, the same pigment that colors human skin, to convert gamma radiation into chemical energy. They're essentially eating radiation, turning one of the most hostile environments humans have created into a fungal buffet. Life finds a way, and fungi write the handbook. They've colonized the International Space Station, growing on surfaces despite efforts to keep the station sterile. They've been found in jet fuel tanks, using hydrocarbons as food. They grow in the Dead Sea, despite salt concentrations that would pickle most organisms. They've even been found growing on the outside of spacecraft, exposed to the vacuum and radiation of space. These extremophile fungi aren't just curiosities. They show us the incredible plasticity of fungal life, the ability to adapt to conditions that push the very boundaries of what we consider habitable. Every extreme environment we explore, we find fungi. They're rewriting our understanding of life's limits, showing us that the possible space for life is far larger than we imagined. The shape-shifting nature of fungi extends beyond individual organisms to the kingdom as a whole. Fungi have evolved forms we're still discovering: rock-eating fungi that mine minerals, carnivorous fungi that trap and consume nematodes, fungi that farm bacteria, fungi that produce light, fungi that generate heat, fungi that create their own air conditioning systems. Each form represents a solution to survival, a way of making a living in a competitive world. The diversity of fungal forms reflects the diversity of ecological niches available on Earth. For every way of obtaining energy and nutrients, there's probably a fungus that's evolved to exploit it. For every environment, no matter how extreme, there's likely a fungus that calls it home. This morphological flexibility, this ability to shapeshift and adapt, might be fungi's greatest strength. While other organisms are locked into specific forms and lifestyles, fungi can transform. They can be single cells or vast networks, parasites or partners, decomposers or producers of new compounds. They can change their shape, their metabolism, their reproductive strategy, their entire way of life in response to environmental changes. In a world where change is the only constant, where environments shift and new challenges constantly arise, the shapeshifters have an advantage. They don't just adapt to change, they embody it. They show us that form follows function, that life is endlessly creative in finding solutions to survival, that the boundaries we draw between different types of organisms are often arbitrary human constructs that nature cheerfully ignores. The fungi remind us that life is far more flexible, far more creative, far more capable of transformation than we often assume. They show us that there are many ways to be alive, many forms that life can take, many solutions to the problems of existence. In their endless shape-shifting, in their morphological creativity, fungi demonstrate that life itself is a shapeshifter, constantly reinventing itself, constantly finding new forms, constantly pushing the boundaries of the possible. The history of human civilization is inseparable from fungi. Long before we understood what they were, before we had microscopes to see their spores or chemistry to analyze their compounds, we were in partnership with these organisms. Beer, bread, and wine civilization, in a glass and on a plate, all depend on fungi. Without them, human history would have taken an entirely different course. Consider ancient Egypt, where beer was so central to life that workers were sometimes paid in beer rations. The pyramids were built by people fueled by fungal fermentation, or ancient Sumeria, where the first written recipe carved into a clay tablet 40 years ago was for beer. The Code of Hammurabi, one of humanity's earliest legal documents, regulated the production and sale of beer. Fungi were literally shaping law and society. Wine, too, has been with us since the beginning of agriculture. Archaeological evidence from Georgia shows wine production from 80 years ago. The ancient Greeks had a god of wine, Dionysus. The Romans spread viticulture across their empire. Medieval monasteries perfected winemaking techniques that we still use today, all of it dependent on Saccharomyces cerevisiae, a single-celled fungus that converts sugar to alcohol. Bread, that most basic of foods, requires yeast to rise. The ancient Egyptians discovered leavened bread around 40 years ago, probably by accident when wild yeasts infected their dough. They called it the food of the gods and developed over 30 different types of bread. The ability to make bread that would keep, that was portable and nutritious, enabled long journeys, military campaigns, the spread of civilization itself. But fermentation was just the beginning. Ancient peoples used fungi in ways we're only now understanding. The ancient Chinese used various mushrooms, medicinally reishi for longevity, shiitake for vigor, cordyceps for stamina. They didn't know about polysaccharides or immunomodulation, but they knew these fungi were special. Our ancestors knew fungi were special, even if they didn't know why. The Greeks and Romans used agaricon, a wood-rotting fungus, to treat consumption, what we now call tuberculosis. Recent analysis shows agaricon does indeed have antimicrobial properties. Indigenous peoples of the Pacific Northwest used the same fungus for spiritual and medicinal purposes. Across the world, separated by vast distances and with no communication, humans discovered the healing power of the same fungi. The Iceman, Ötzi, found frozen in the Alps and dating from 300 years ago, carried two types of fungi with him. One was amadou, useful for starting fires. The other was birch polypore, which has antimicrobial and antiparasitic properties. He was carrying a fungal first aid kit, 30 years before the first written medical texts. The transformation of fungi from folk medicine to modern pharmaceuticals began with one of the most famous accidents in scientific history. In 1928, Alexander Fleming returned from vacation to find that a petri dish of Staphylococcus bacteria had been contaminated with mold. Instead of throwing it away, he noticed something extraordinary. The bacteria around the mold were dead. The mold, Penicillium notatum, was producing something that killed bacteria. Fleming's discovery might have remained a laboratory curiosity, if not for World War II. The urgent need for antibiotics to treat wounded soldiers drove massive investment in penicillin production. Howard Florey and Ernst Chain figured out how to mass-produce penicillin using deep tank fermentation methods that are still used today. By D-Day, the Allies had enough penicillin to treat all wounded soldiers. A contaminated Petri dish had changed the course of medicine and saved millions of lives. Penicillin was just the beginning of fungi's pharmaceutical revolution. Today, fungi give us statins, the cholesterol-lowering drugs taken by millions. Lovastatin, the first statin, comes from Aspergillus terreus. These drugs have prevented countless heart attacks and strokes, adding years to millions of lives. Cyclosporine, derived from the fungus Tolypocladium inflatum, made organ transplantation possible. Before cyclosporine, the immune system would reject transplanted organs. This fungal compound selectively suppresses immune responses, allowing kidneys, hearts, livers, and other organs to be successfully transplanted. Thousands of people are alive today because of a compound from a fungus found in a soil sample from Norway. Modern pharmaceutical fungi are yielding new medicines at an accelerating pace. Fungi produce compounds that fight cancer: paclitaxel from endophytic fungi, lentinan from shiitake mushrooms, PSK from turkey tail fungi. These aren't alternative medicines, but FDA-approved drugs used in mainstream cancer treatment. A pharmacy growing on fallen logs and in forgotten corners, waiting to be discovered. Fungi are also producing entirely new classes of antibiotics, crucial as bacterial resistance to existing drugs grows. Pleuromutilins from the fungus Pleurotus mutilus represent the first new class of antibiotics in decades. As we face the spectre of antibiotic-resistant superbugs, fungi may once again save us from bacterial diseases. The industrial applications of fungi extend far beyond medicine. Fungi are enzyme factories, producing biological catalysts that industry desperately needs. Your laundry detergent probably contains fungal enzymes that break down protein stains. Your jeans might have been treated with fungal cellulases to give them that soft, worn feel. The bread you eat uses fungal amylases to break down starches. Citric acid, which gives soft drinks their tang and acts as a preservative in countless foods, comes from Aspergillus niger. We produce over 2 million tons of citric acid annually, almost all from fungal fermentation. It would take the juice of billions of lemons to produce the same amount from citrus fruits. Fungi do it from simple sugars, cheaply and efficiently. Industrial enzymes from fungi are replacing harsh chemical processes. Fungal lacases can bleach paper and textiles without chlorine. Fungal lipases can produce biodiesel from waste oils. Fungal proteases can recover silver from X-ray films. Each enzyme represents a chemical process that occurs at room temperature, in water, without toxic byproducts. Fungi as factories, but factories that improve the environment rather than pollute it. The future of fungal biotechnology is even more extraordinary. Mycelium, the root-like network of fungal hyphae, is being developed as a building material. Companies are growing mycelium into specific shapes: bricks, panels, packaging materials. Feed agricultural waste to certain fungi, and they'll digest it and bind it together with their mycelium, creating materials that are strong, light, fire-resistant, and completely biodegradable. These mycelium materials can replace plastics, styrofoam, even concrete in some applications. Imagine buildings with walls grown from fungi, packaging that decomposes in your garden, furniture that's literally alive while you're using it. Major companies are already using mycelium packaging. Adidas has produced shoes with mycelium leather. The future of materials might be grown rather than manufactured. Even more remarkably, researchers are exploring fungi as living computers. Fungal networks can process information, with electrical signals traveling through mycelium in ways reminiscent of neural networks. Scientists have created fungal circuits that can perform basic computational tasks. These biological computers could self-repair, self-power, and self-replicate. Perhaps the future isn't silicon and steel, but soft and living. Fungi are being engineered to produce everything from vanilla flavoring to spider silk proteins. They can create chemicals that would be impossible or prohibitively expensive to synthesize artificially. They're being developed to produce biofuels from agricultural waste, to create new plastics that truly biodegrade, to manufacture complex pharmaceuticals that currently require dozens of chemical steps. The potential of fungal biotechnology seems limitless. Fungi that can detect and neutralize chemical weapons. Fungi that can extract rare earth elements from electronic waste. Fungi that can create quantum dots for next-generation solar panels. Every month brings new discoveries, new applications, new possibilities. But with great power comes great responsibility. As we harness fungi's incredible capabilities, we must consider what we owe to organisms that give us so much. The history of human exploitation of nature is littered with extinctions, ecosystem collapses, and unintended consequences. We cannot afford to repeat these mistakes with fungi. Many medicinal mushrooms are already threatened by overharvesting. Cordyceps sinensis, the caterpillar fungus prized in traditional Chinese medicine, has become so valuable that it's causing social and environmental problems in the Himalayas. People are fighting over collection rights, and the fungus is becoming increasingly rare in the wild. Pacific Northwest forests are being damaged by commercial harvesting of matsutake and chanterelle mushrooms. Harvesters sometimes rake the forest floor, destroying mycelium and preventing future fruiting. Old-growth forests, which produce the most valuable mushrooms, are under pressure from both logging and mushroom harvesting. Sustainable harvesting and cultivation must be our priority. We need to learn to grow more fungi in controlled conditions rather than depleting wild populations. Many valuable fungi can now be cultivated: shiitake, oyster mushrooms, lion's mane, and others. But some, like truffles and porcini, still resist cultivation, maintaining their mysterious requirements for specific trees and soils. We also need to consider the ethics of genetic modification of fungi. As we engineer fungi to produce new compounds or perform new functions, we're creating organisms that have never existed before. What are our responsibilities to these new life forms? What are the risks of release into the environment? These are questions we must answer carefully, thoughtfully, with full consideration of long-term consequences. Taking only what we need, giving back what we can, this should be our guiding principle. For every antibiotic we derive from fungi, we should protect fungal habitats. For every industrial application we develop, we should ensure it truly reduces environmental impact. For every new fungal technology we create, we should consider its full life cycle and ultimate fate. Indigenous peoples have long understood the importance of reciprocity with nature. Many cultures have traditions of giving thanks to mushrooms before harvesting, of taking only what's needed, of ensuring the continued health of fungal populations. We could learn from these approaches, combining traditional wisdom with modern science. The relationship between humans and fungi is ancient and ongoing. From the first fermented beverages to the latest biotechnologies, fungi have been our partners in creating civilization. They've fed us, healed us, inspired us, and increasingly, they're showing us new ways to live sustainably on this planet. As we face challenges like climate change, antibiotic resistance, and plastic pollution, fungi offer solutions. They can break down our waste, create new materials, produce our medicines, even help us explore space. Researchers are investigating, using fungi to create habitats on Mars, growing living buildings from Martian soil. The alchemists of old sought to transform base metals into gold. Fungi do something far more valuable. They transform waste into resources, poisons into medicines, problems into solutions. They are the true alchemists, and we're only beginning to understand their secrets. Our future is intertwined with fungi in ways we're only starting to appreciate. As we learn more about these remarkable organisms, as we develop new technologies based on their capabilities, we must remember that we're not masters, but partners. Fungi have been here far longer than us, and will likely be here long after we're gone. Our wisdom lies not in controlling them, but in learning from them, working with them, and respecting them. The story of fungi in human history is far from over. Each discovery opens new possibilities, new applications, new appreciations for these organisms that have shaped our past and will undoubtedly shape our future. From the bread on our tables to the medicines in our pharmacies, from the materials of tomorrow to the computers of the future, fungi are and will remain the alchemists among us, transforming our world in ways both visible and invisible, known and yet to be discovered. In recognizing fungi as the biochemical wizards they are, we recognize that nature has already solved many of the problems we're struggling with. The solutions are there, growing on fallen logs, fruiting in forest shadows, spreading through soil in invisible networks. We just need the wisdom to see them, the patience to understand them, and the humility to learn from organisms that have been practicing chemistry for 1 billion years longer than we've existed. Among all of fungi's remarkable properties, perhaps none is more mysterious and controversial than their ability to alter human consciousness. Certain mushrooms contain compounds that profoundly change perception, emotion, and thought substances that let us peek behind the curtain of ordinary perception. These psychedelic fungi have been intertwined with human culture for millennia, serving as sacraments, medicines, and doorways to other ways of seeing the world. The archaeological evidence for human use of psilocybin mushrooms stretches back thousands of years. In the Sahara Desert, rock art from 7,090 years ago depicts what appear to be mushroom-headed figures and mushrooms sprouting from human bodies. In Spain, the Selva Pascuala cave mural from 60 years ago seems to show Psilocybe hispanica mushrooms. These weren't casual illustrations, they were important enough to paint on sacred cave walls, suggesting these fungi held deep significance for our ancestors. In Mesoamerica, the evidence becomes even clearer. The Maya and Aztec civilizations revered psilocybin mushrooms, calling them Teonanacatl, flesh of the gods. Stone mushroom effigies dating from 500 BCE to 900 CE have been found throughout Guatemala and Mexico. The Florentine Codex, compiled in the 16th century, describes Aztec mushroom ceremonies in detail. These weren't recreational experiences but profound religious sacraments, carefully controlled and deeply respected. The Mazatec people of Oaxaca, Mexico, maintained this sacred mushroom tradition through centuries of suppression. When Spanish colonizers banned indigenous religious practices, including mushroom use, the Mazatec continued their ceremonies in secret. They preserved not just the knowledge of which mushrooms to use, but the entire ceremonial context, the songs, the prayers, the healing practices that had been refined over generations. It was a Mazatec curandera, Maria Sabina, who in 1955 introduced psilocybin mushrooms to R. Gordon Wasson, a banker and amateur mycologist. Wasson's subsequent article in Life magazine, Seeking the Magic Mushroom, introduced psychedelic mushrooms to Western consciousness. What followed was a cultural earthquake whose reverberations we're still feeling today. Indigenous knowledge of psychedelic fungi extends far beyond Mesoamerica. In Siberia, shamans used Amanita muscaria, the iconic red and white spotted mushroom, in their spiritual practices. The Sami people of northern Europe had similar traditions. These practices weren't about escaping reality, but about accessing different layers of it, about healing, divination, and maintaining the balance between human and more than human worlds. What these indigenous traditions understood intuitively, modern science is now explaining neurologically. Psilocybin, the primary psychedelic compound in magic mushrooms, is remarkably similar in structure to serotonin, one of our brain's key neurotransmitters. When consumed, psilocybin is converted to psilocin, which binds to serotonin receptors, particularly the 5-HT2A receptor, triggering cascades of neural activity that dramatically alter consciousness. Brain imaging studies have revealed something extraordinary about how psilocybin affects neural activity. Rather than increasing brain activity as you might expect from such profound experiences, psilocybin actually decreases activity in certain key areas, particularly in what's called the default mode network, DMN. The DMN is a network of brain regions that's active when we're not focused on the outside world, when we're thinking about ourselves, remembering the past, planning the future, or engaging in self-referential thought. The DMN is sometimes called the me network because it's centrally involved in our sense of self, our ego, our feeling of being separate individuals distinct from the world around us. Under psilocybin, this network becomes less active and less coordinated. It's like defragmenting the hard drive of consciousness, temporarily dissolving the patterns that usually organize our thoughts and perceptions. As the default mode network quiets, other brain networks that don't usually communicate start talking to each other. Visual processing areas connect with auditory regions. Emotional centers communicate with areas usually devoted to abstract reasoning. The brain becomes, in a sense, more integrated and less segregated. This increased connectivity might explain the synesthesia many people experience on psychedelics, seeing sounds, hearing colors, feeling thoughts as physical sensations. This temporary dissolution of normal brain networks has profound therapeutic implications. Depression, anxiety, PTSD, and addiction all involve rigid patterns of thought and feeling mental ruts that people can't escape. Psychedelics appear to temporarily flatten the mental landscape, allowing new patterns to form. It's not that the drugs themselves cure these conditions, but they create a window of neuroplasticity during which therapeutic change becomes more possible. The clinical research on psilocybin therapy is remarkably promising. Studies at Johns Hopkins, NYU, and Imperial College London have shown that 1 or 2 psilocybin sessions, combined with therapy, can produce lasting improvements in treatment-resistant depression. Patients describe the experience as being like years of therapy compressed into a few hours. They report seeing their problems from new perspectives, feeling connected to something larger than themselves, experiencing profound self-compassion for the first time. For patients with terminal cancer facing end-of-life anxiety, psilocybin therapy has been transformative. In clinical trials, a single psilocybin session reduced death anxiety and depression in about 80% of participants, with benefits lasting months or even years. Patients describe losing their fear of death, feeling connected to a continuity of existence that transcends their individual lives. One participant said it was like, "The universe opened up and showed me that everything is connected and eternal." PTSD, that cruel disorder that traps people in loops of traumatic memory, also appears responsive to psychedelic therapy. Early trials combining MDMA, not a fungus but working through similar mechanisms with therapy, have shown remarkable success. Psilocybin trials for PTSD are underway, with similar promise. The medicine seems to allow people to revisit traumatic memories without being overwhelmed by them, to integrate these experiences rather than being haunted by them. Addiction, too, is yielding to psychedelic therapy. Johns Hopkins researchers found that psilocybin therapy helped long-term smokers quit at rates far exceeding any existing treatment. Alcoholism, cocaine addiction, and other substance use disorders are all being studied. The psychedelics seem to interrupt the loops of craving and reward, allowing people to see their addictions from outside the prison of dependent thinking. Sometimes healing means temporarily dissolving the boundaries we've built. Boundaries between self and trauma, between individual and community, between human and nature. Psychedelics facilitate this dissolution, not as an escape from reality, but as a reconnection with aspects of reality our normal consciousness filters out. The philosophical implications of psychedelic experiences have fascinated thinkers since they first entered Western consciousness. What does it mean that a simple molecule from a mushroom can so profoundly alter our perception of reality? What does this say about the nature of consciousness itself? One perspective is that consciousness is far more flexible and multiple than we usually realize. Our ordinary waking consciousness, with its sharp subject-object distinctions and linear thought patterns, is just one possible configuration of mind. Psychedelics reveal other configurations, ones where boundaries dissolve, where time becomes fluid, where the universe feels alive and communicating. These aren't hallucinations in the dismissive sense, but rather different ways of processing the same reality. The experience of ego dissolution under psilocybin raises profound questions about the self. If the self can dissolve and reform, if we can experience consciousness without the usual boundaries of me and not me, what does this say about the nature of identity? Many people report that psychedelic experiences show them that the separate self is a construction, useful for navigating daily life but not the deepest truth of what we are. The feeling of interconnection that psychedelics often produce, the sense that all life is one, that separation is an illusion, aligns remarkably with both ancient spiritual teachings and modern ecology. Perhaps mushrooms are Earth's way of teaching us we're not separate from nature. After all, these fungi have been part of ecosystems for hundreds of millions of years. When we ingest them, we're literally taking the forest into our bodies, allowing its chemistry to restructure our consciousness. Some researchers and philosophers have proposed that psychedelic experiences reveal something true about the nature of reality, that consciousness is fundamental, that everything is interconnected, that the universe is more alive and aware than our ordinary perception suggests. Others argue these are simply drug-induced states with no particular truth value. The debate continues, but what's undeniable is that these experiences often produce lasting changes in people's values, behaviors, and worldviews. The mystical experience, occasioned by psilocybin, characterized by unity, transcendence of space and time, ineffability, and a noetic quality of revealed truth, appears to be the key therapeutic factor. In studies, the strength of the mystical experience correlates with positive therapeutic outcomes. It's not just brain chemistry, but the content of the experience, the insights and revelations that produce healing. However, we must approach these powerful substances with appropriate caution and respect. These aren't toys, but tools, ancient and demanding respect. The same experiences that can heal can also harm if approached carelessly. Bad trips, though less common in controlled therapeutic settings, can be terrifying and occasionally trigger lasting psychological distress. Setting your mindset, going into the experience and the environment in which it occurs, are crucial. Indigenous cultures understood this, embedding psychedelic use in elaborate rituals with experienced guides. Modern therapeutic protocols similarly emphasize preparation, a safe and supportive environment, and integration afterward to make sense of the experience. The importance of integration cannot be overstated. A psychedelic experience, no matter how profound, is just the beginning. The real work comes in integrating insights into daily life, in making concrete changes based on what was revealed. Without integration, these experiences can become just another form of spiritual bypassing, a way to feel enlightened without doing the hard work of actual transformation. There are also important physiological considerations. People with certain heart conditions, schizophrenia, or bipolar disorder should not use psychedelics. These substances can trigger psychotic episodes in predisposed individuals. They can interact dangerously with certain medications. They're not for everyone, and they're certainly not casual party drugs. The current psychedelic renaissance in medicine and culture requires us to learn from past mistakes. The 1960s saw psychedelics burst into Western culture without adequate frameworks for safe use, leading to casualties and backlash that set research back decades. This time, we need to proceed more carefully, respecting both the power of these substances and the wisdom of cultures that have used them for millennia. Legal and ethical questions abound. Should psychedelics be available only in medical settings or also for personal growth and spiritual exploration? How do we prevent commercialization from corrupting these medicines? How do we honor indigenous traditions while making these tools available to those who might benefit? These are questions our society is just beginning to grapple with. What's clear is that psychedelic fungi offer unique tools for understanding consciousness, treating mental illness, and potentially transforming our relationship with nature and each other. They remind us that consciousness is far more mysterious and mutable than we often assume. They show us that healing sometimes requires not adding something new but dissolving something old. Old traumas, old patterns, old ways of seeing that no longer serve us. As we continue to study these remarkable fungi, we're not just learning about mushrooms or even about medicine. We're learning about the nature of mind, the possibilities of healing, and our place in the larger web of life. The mind-bending properties of certain fungi are teaching us that our minds are far more flexible than we thought, that consciousness is stranger and more wonderful than we imagined, and that sometimes the medicine we need has been growing in the forest all along, waiting patiently for us to be ready to receive its teachings. Lichens represent one of evolution's most successful partnerships, a marriage so successful it created a new form of life. For the longest time, scientists couldn't figure out what lichens were. They seemed like plants, but had no roots, leaves, or flowers. They grew in impossible places where no plant could survive. It wasn't until the 1860s that Swiss botanist Simon Schwendener proposed something radical. Lichens weren't single organisms, but partnerships between fungi and algae. The scientific community ridiculed Schwendener's idea. The notion that two entirely different organisms could merge so completely that they became indistinguishable as separate entities seemed absurd. Yet he was right, and if anything, he underestimated the complexity. We now know that many lichens involve not just two partners but three or more multiple fungi, algae, and bacteria living in intimate association. The basic lichen partnership is elegantly simple. The fungus provides structure, protection, and minerals. The alga, or cyanobacterium, provides sugars from photosynthesis. Together, they can survive where neither could alone on bare rock, in Arctic tundra, in deserts, on tree bark, even on gravestones and old buildings. There are over 20 known lichen species, representing multiple independent evolutionary origins of this partnership strategy. Lichens are the pioneers, the first visible life to colonize bare rock after glaciers retreat or volcanoes create new land. They're writing the first drafts of ecosystems, beginning the slow process of turning stone into soil. A lichen can take decades to grow a few centimeters, but it can live for thousands of years, slowly, patiently breaking down rock and building the foundation for other life. The way lichens colonize bare rock is a masterclass in biochemical engineering. The fungal partner produces acids that slowly dissolve minerals from the rock surface. These minerals feed both partners and, when the lichen dies, become part of the first proto-soil. The fungal hyphae penetrate microscopic cracks in the rock, gradually widening them through expansion and contraction with wet-dry cycles. Some lichens can survive losing 98% of their water content, entering a state of suspended animation until moisture returns. Recent discoveries have made lichens even more remarkable. In 2016, scientists discovered that many lichens contain not just one fungal partner, but a second yeast that lives in the outer cortex. This yeast, undetected for 150 years of lichen study, appears crucial for creating the chemicals that give different lichens their distinctive colors and properties. Some lichens may involve dozens of microbial partners, each contributing something essential to the collective. Lichens produce over 10 unique chemical compounds, many found nowhere else in nature. These chemicals protect against UV radiation, prevent other organisms from growing on them, and possibly serve functions we haven't yet discovered. The antibiotic properties of lichen compounds have been used in traditional medicine for millennia. Modern research is finding that lichen chemicals might fight antibiotic-resistant bacteria, cancer cells, and viruses. The lichen symbiosis is so integrated that they reproduce as a unit. While the fungal and algal partners can theoretically reproduce separately, most lichen reproduction involves structures that contain both partners. Soredia, tiny balls of fungal hyphae wrapped around algal cells, break off and disperse to found new lichen colonies. It's like dispersing preformed partnerships, ensuring the symbiosis continues. But the symbiotic dance extends far beyond lichens. Inside the leaves and stems of virtually every plant on Earth live endophytic fungi. Fungi that colonize plant tissues without causing disease. A single tree might host hundreds of endophytic fungal species. A single leaf might contain dozens. These fungi are so ubiquitous that finding a plant without them is exceptional. For decades, botanists ignored endophytic fungi or considered them latent pathogens waiting for the plant to weaken. We now understand they're bodyguards you can't see, working at the cellular level. They produce chemicals that deter herbivores, fight off pathogenic fungi and bacteria, and even help plants tolerate drought and heat stress. The relationship between endophytic fungi and plants blurs the line between organism and environment. Some endophytes are passed from parent to offspring through seeds, becoming part of the plant's inheritance. Others must be acquired from the environment each generation. Some can switch between endophytic and free-living lifestyles depending on conditions. Certain grass endophytes produce alkaloids that make the grass toxic to herbivores, but not to the plant itself. Livestock that eat endophyte-infected fescue can suffer from fescue toxicosis. But the grass thrives because it's protected from grazing. The fungus gets shelter and nutrients. The grass gets chemical defenses it couldn't produce on its own. In tropical forests, endophytic fungi and leaves create a chemical landscape of incredible diversity. Each leaf becomes a patchwork of different fungal territories, each producing different chemicals. This diversity might explain why tropical forests, despite their dense vegetation, don't suffer from devastating disease epidemics as often as monoculture crops do. The endophytes create a kind of herd immunity at the chemical level. Some endophytes provide more dramatic services. The fungus Curvularia protuberata allows its host grass to grow in Yellowstone's geothermal soils, where temperatures can reach 60 degrees. Without the fungus, the grass dies. Without the grass, the fungus can't survive the heat either. Together, they thrive in conditions that would kill either partner alone. Even more remarkably, this heat tolerance can be transferred. Infect a tomato plant with this fungus, and it too becomes heat tolerant. The animal kingdom has evolved equally remarkable fungal partnerships. Leafcutter ants represent agriculture invented millions of years before humans. These ants don't eat the leaves they cut, they feed them to fungal gardens they maintain in underground chambers. The fungus breaks down the tough plant material into nutrients the ants can digest. This ant-fungus agriculture is astoundingly sophisticated. The ants maintain specific fungal cultivars that have been propagated for millions of years. When a queen ant leaves to found a new colony, she carries a piece of the fungus in a special pocket in her mouth. The ants weed their gardens, removing competing fungi and bacteria. They apply antibiotics produced by bacteria living on their bodies, a kind of biological pesticide that targets garden pests but not their crop. The fungus, for its part, has evolved special structures called gongilidia, nutrient-rich swellings that exist solely to feed the ants. These structures appear in no other context, only in ant gardens. The fungus has become so specialized for ant cultivation that it can no longer survive in the wild. This is obligate mutualism, both partners completely dependent on each other. Different leafcutter ant species cultivate different fungi, and the complexity of their agriculture varies. Some species are generalists, growing fungi that can still survive outside ant colonies. Others are specialists with fungi that exist nowhere else. The most derived species have evolved industrial-scale agriculture, with colonies of millions of ants maintaining fungal gardens the size of small rooms. Termites have independently evolved their own form of fungal agriculture. Macrotermitine termites in Africa and Asia cultivate termitomyces fungi in underground gardens. But these termites have taken fungal partnership even further. The metabolic heat from their fungal gardens helps regulate nest temperature and humidity. The fungi are essentially living air conditioners, maintaining optimal conditions for the termite colony. The termite fungus system creates some of nature's most impressive structures. The towering termite mounds of Africa, some over 30 feet tall, are ventilation systems for underground fungal gardens. Air flows through the mound's channels, regulated by the opening and closing of vents, maintaining the precise conditions the fungus needs. The entire structure, termites, fungus, and mound functions as a superorganism with emergent properties none of the components possess alone. Perhaps the most intimate fungal partnership is the one we carry within ourselves. The human gut microbiome, the fungal community in our intestines, is only beginning to be understood. We harbor dozens of fungal species, with Candida, Saccharomyces, and Malassezia being the most common. You're never alone, you're a walking ecosystem. The gut microbiome interacts with our bacterial microbiome, our immune system, and possibly our nervous system. Changes in gut fungi have been associated with inflammatory bowel disease, allergies, and even neurological conditions. Some researchers suggest that gut fungi might influence our food cravings, our mood, even our behavior, though this remains controversial. Candida albicans, present in most humans, exemplifies the complexity of our fungal relationships. In healthy individuals, it exists harmlessly in the gut, but if the bacterial microbiome is disrupted by antibiotics, for instance, Candida can overgrow, causing infections. The same organism can be commensal, beneficial, or pathogenic depending on context. The boundary between friend and foe is constantly negotiated through chemical signaling we're only beginning to understand. We're also discovering that we share fungi with those around us. Couples who live together develop similar microbiomes. People who share spaces exchange fungi constantly. Our fungal communities are shaped by our pets, our homes, our diets, our medications. We're not individuals but communities, and those communities extend beyond our bodies to include those we live with. The spectrum from parasitism to mutualism reveals how fluid these relationships can be. Many relationships we call parasitic might have mutual benefits we haven't recognized. Some fungi that parasitize insects also protect them from other pathogens. Some plant pathogens increase their host's competitive ability against other plants. The labels we use, parasite, commensal, mutualist, are human constructs that don't capture the full complexity of these interactions. Evolution shows us that today's parasite might be tomorrow's partner. Mitochondria, the powerhouses of our cells, were once free-living bacteria that were engulfed by ancient cells. What might have started as parasitism or predation became the most fundamental partnership in complex life. Many current symbioses probably began as antagonistic relationships that evolved toward mutual benefit. The fig wasp and fig tree relationship illustrates this evolutionary dance. Fig wasps lay their eggs only in fig flowers, and fig trees can only be pollinated by their specific wasp species. This extreme mutual dependence probably evolved from a more antagonistic relationship, where wasps parasitized figs without providing benefits. Over millions of years, the relationship evolved toward mutualism, with both partners now completely dependent on each other. The orchid mycorrhizal relationship shows another pathway to partnership. Orchid seeds are tiny dust-like particles with no nutrient reserves. They can't germinate without fungal partners to provide nutrients. Young orchids are essentially parasites on their fungi, but as they mature and begin photosynthesizing, the relationship becomes more reciprocal. Some orchids remain cheaters their whole lives, never giving back to their fungal partners. Others become generous partners, sharing abundant sugars. The same fungal species might simultaneously be parasitized by one orchid and partnering mutually with another. These fluid relationships suggest that symbiosis isn't a fixed state but a dynamic negotiation Partners constantly adjust their investment based on costs and benefits. A fungus might provide more nutrients to a plant when carbon is abundant, but withhold them when carbon is scarce. A plant might feed fungal partners when it needs minerals but reduce payment when minerals are plentiful. Environmental conditions can shift relationships along the mutualism-parasitism spectrum. The same fungus that helps a plant survive drought might become parasitic in wet conditions. Climate change is likely reshuffling these relationships in ways we can't yet predict. Some mutualisms might become parasitisms. Some parasitisms might become mutualisms. The symbiotic dance also involves chemical communication of stunning complexity. Plants and fungi exchange hundreds of chemical signals, negotiating terms of trade, warning of dangers, coordinating responses to stress. These chemical conversations happen at speeds and scales we're only beginning to measure. A single root hair might be simultaneously communicating with dozens of fungal species, each conversation mediated by different chemical languages. Some fungi manipulate their partners through chemical signaling. They can make plants more generous with carbon payments by mimicking plant hormones. They can suppress plant immune responses that would otherwise limit colonization. They can even alter plant behavior, making roots grow toward them, making leaves produce chemicals that benefit the fungus. The line between communication and manipulation is often unclear. The evolutionary implications of symbiosis are profound. Lynn Margulis argued that symbiosis, not just competition, drives evolution. Major transitions in evolution, the origin of eukaryotic cells, the colonization of land by plants, the evolution of herbivory in animals, all involved symbiosis with microbes, including fungi. We are not the products of singular evolutionary lineages, but of mergers, acquisitions, and partnerships accumulated over billions of years. This view radically changes how we think about organisms. Are we individuals or communities? Where does the plant end and the fungus begin when they share nutrients, chemical defenses, even genetic material? These questions aren't just philosophical, they have practical implications for agriculture, medicine, and conservation. Understanding symbiosis is revealing new approaches to agriculture. Instead of sterilizing soil and adding chemical inputs, we might manage soil microbial communities. Instead of breeding crops in isolation, we might breed plant-fungal partnerships. Instead of seeing diseases as invasions to repel, we might see them as dysbiosis to rebalance. The symbiotic dance between fungi and other organisms shows us that life is fundamentally collaborative. Competition exists, but it operates within a framework of cooperation. The most successful organisms aren't necessarily the strongest competitors, but the best collaborators, the ones that form and maintain beneficial partnerships. As we face global challenges that require unprecedented cooperation, fungi and their partners offer lessons. They show us that different organisms with different capabilities can create emergent properties neither possesses alone. They demonstrate that successful partnerships require constant negotiation and adjustment. They prove that diversity strengthens resilience, that networks are more robust than hierarchies, that sharing resources can benefit all parties. The symbiotic dance continues all around us, in us, through us. Every breath we take is made possible by ancient partnerships. Every step we take is on soil created by symbiosis. We are not observers of this dance, but participants. Our bodies hosting fungal partners, our agriculture depending on soil symbiosis, our future intertwined with the networks of cooperation that fungi have been weaving for 1 billion years. Fungi are nature's most creative chemists, producing an estimated 150 different secondary metabolites, compounds not essential for basic growth, but serving specialized functions. To put this in perspective, all of modern pharmaceutical chemistry has produced perhaps 20 distinct compounds. Fungi have been at this far longer, with far more sophistication, creating molecules of staggering complexity, without test tubes, without laboratories, without any of the tools we consider essential for chemistry. Secondary metabolites are where fungi truly show their chemical genius. Unlike primary metabolites, the sugars, amino acids, and lipids needed for basic life, secondary metabolites are optional, experimental, creative. They're antibiotics, toxins, pigments, signaling molecules, compounds that glow in the dark, compounds that smell like almonds or rotting flesh, compounds that can cure disease or cause hallucinations. They're running millions of tiny chemistry experiments all the time, each species, even each strain, producing its own unique chemical signature. The diversity is mind-boggling. Aspergillus nidulans, a common mold, has 32 gene clusters for secondary metabolite production, capable of producing dozens of different compounds. But here's the remarkable part. Under normal laboratory conditions, most of these gene clusters are silent. The fungus has the genetic machinery to produce these compounds but doesn't. Change the conditions, add a competitor, alter the pH, limit nutrients, and suddenly new compounds appear. It's as if fungi carry a vast library of chemical recipes, pulling out different ones depending on circumstances. We've likely discovered less than 5% of fungal secondary metabolites. Every year, researchers identify thousands of new fungal compounds, many with properties unlike anything in our chemical databases. There are antifungals that work by mechanisms we don't understand, anti-cancer compounds that target pathways we didn't know existed, antibiotics that bacteria haven't developed resistance to because they've never encountered them. Some secondary metabolites are so complex that we still can't synthesize them artificially, despite knowing their structure. Taxol, originally from yew trees but actually produced by an endophytic fungus, required decades of work to synthesize. Many fungal compounds have multiple chiral centers, three-dimensional structures that must be exactly right for biological activity. Fungi assemble these molecules with casual precision, at room temperature, in water, without any of the harsh chemicals or conditions our synthesis methods require. Beyond their role as chemists, fungi are the accountants of Earth's elemental economy, managing the flow of carbon, nitrogen, phosphorus, and other elements through ecosystems. Without fungi, these biogeochemical cycles would collapse, and life as we know it would be impossible. The carbon cycle depends critically on fungi. They're responsible for decomposing much of the 100 billion tons of plant material produced annually. Without fungal decomposition, carbon would accumulate in dead plant matter, eventually depleting atmospheric CO2 and ending photosynthesis. Fungi regulate the balance between carbon storage and release, acting as both carbon sinks and sources depending on conditions. Mycorrhizal fungi are particularly important for carbon cycling. They receive 5–20% of all carbon fixed by plants through photosynthesis, a massive flow of carbon from atmosphere to soil. Some of this carbon feeds the fungi, but much is converted to glomalin, a glycoprotein that can persist in soil for decades. Glomalin accounts for 30% of carbon in some soils, storing more carbon than the atmosphere contains. Climate change is altering fungal carbon cycling in ways we're still trying to understand. Warming temperatures accelerate decomposition, potentially releasing stored carbon, but warming also increases plant growth and carbon flow to mycorrhizal fungi, potentially increasing storage. The balance between these processes will significantly influence future atmospheric CO2 levels. Nitrogen cycling is equally dependent on fungi. While bacteria get credit for nitrogen fixation, fungi play crucial roles in making nitrogen available to plants. They decompose proteins and nucleic acids, releasing ammonia. They compete with bacteria for nitrogen, regulating its availability. Mycorrhizal fungi can access organic nitrogen sources that plants can't use directly, effectively expanding the nitrogen pool available to ecosystems. Fungi also produce and consume nitrous oxide, a greenhouse gas 300 times more potent than CO2. Some fungi reduce N2O to harmless N2. Others produce it during decomposition. The balance between these processes affects atmospheric N2O levels and climate. As we alter nitrogen cycles through fertilizer use, we're changing fungal communities in ways that could amplify or mitigate greenhouse gas emissions. Phosphorus, essential for all life, but often limiting in ecosystems, is mobilized primarily by fungi. Unlike nitrogen, which has a gaseous phase, phosphorus cycles only through solid and dissolved forms. Fungi produce phosphatase enzymes that release phosphorus from organic matter. Mycorrhizal fungi extend plant access to phosphorus by exploring soil volumes roots can't reach, and by producing organic acids that dissolve mineral phosphorus. The remediation powers of fungi are perhaps their most immediately practical chemical capability. Mycoremediation using fungi to clean up pollution works because fungi evolved to break down complex organic molecules. The same enzymes that decompose lignin can break down petroleum products, pesticides, even explosives. Nature's cleanup crew, working one molecule at a time. Petroleum products, those complex hydrocarbons we've pulled from deep underground and spread across Earth's surface, are surprisingly vulnerable to fungal attack. Oyster mushrooms can break down diesel fuel and motor oil. Turkey tail fungi can degrade PAHs polycyclic aromatic hydrocarbons, carcinogenic compounds in petroleum. Some fungi can even break down heavy crude oil, those tar-like substances that persist for decades after spills. The mechanisms are elegant. Fungi produce extracellular enzymes, lignin peroxidases, manganese peroxidases, laccases that generate highly reactive free radicals. These radicals attack complex molecules almost indiscriminately, breaking carbon-carbon bonds, adding oxygen, transforming hydrophobic compounds into water-soluble ones that other organisms can further degrade. It's controlled combustion at the molecular level. Plastic degradation by fungi is an emerging field with enormous potential. Pestaloteuthis microspora, discovered in the Amazon, can break down polyurethane in anaerobic conditions. Aspergillus tuberculosis can degrade plastics in weeks that would otherwise persist for decades. These fungi are producing enzymes we didn't know existed, targeting bonds we thought were biologically inert. The challenge with mycoremediation isn't getting fungi to break down pollutants. Many fungi can do this naturally. The challenge is optimization. Finding the right fungi for specific pollutants, maintaining them in contaminated environments, ensuring complete breakdown rather than partial transformation into toxic intermediates. It's not enough to break down a pesticide if the breakdown products are more toxic than the original. Heavy metal remediation presents different challenges. Fungi can't break down metals, but they can transform them into less toxic forms or concentrate them for removal. Some fungi precipitate metals as oxalates or carbonates. Others absorb metals into their cells, concentrating them from large volumes of contaminated soil or water into small volumes of fungal biomass that can be harvested and properly disposed of. Chemical communication is perhaps the most sophisticated aspect of fungal chemistry. Fungi produce and detect hundreds of volatile organic compounds, VOCs, creating an invisible chemical landscape of signals and responses. These aren't random emissions, but precisely regulated messages carrying specific information. When fungi are attacked by bacteria, they release VOCs that attract predatory bacteria that eat their attackers. When competing with other fungi, they produce VOCs that inhibit their competitors' growth. Some fungi produce VOCs that smell like flowers to attract insects for spore dispersal. Others produce VOCs that smell like rotting meat for the same purpose. The diversity of strategies is remarkable. The smell of rain petrichor is partly produced by fungi. When rain hits dry soil, it releases fungal and bacterial VOCs that have accumulated during dry periods. Geosmin, produced by various soil fungi and bacteria, is detectable by humans at concentrations of 5 parts per trillion. We can smell it better than sharks can smell blood. This sensitivity might have evolved because rain and the associated fungal activity signal good conditions for finding food or fertile soil for agriculture. Fungi use VOCs for long-distance communication in ways we're only beginning to understand. Trichoderma fungi produce 6-pentyl alpha, pyrone, which smells like coconut to us but signals enemy approaching to other fungi. Some mycorrhizal fungi produce VOCs that make their host plants more attractive to beneficial insects, or less attractive to herbivores. It's a chemical language we're only beginning to translate. The complexity of fungal chemical communication becomes even more apparent when we consider that the same compound can have different meanings in different contexts. A VOC that attracts one insect might repel another. A compound that signals food here at low concentrations might signal danger at high concentrations. The meaning depends on the receiver, the concentration, the presence of other compounds, the environmental context. Some fungi manipulate other organisms through chemical signals with remarkable precision. Ophiocordyceps fungi, which parasitize insects, produce compounds that manipulate their host's behavior. Infected ants climb to specific heights and bite onto leaves with a death grip, positioning themselves perfectly for fungal spore dispersal. The fungi are producing neuroactive compounds that hijack the ant's nervous system, but we still don't understand exactly how this works. Fungi also produce compounds that affect plant behavior. Some endophytic fungi produce auxin-like compounds that alter plant growth. Others produce gibberellins that affect plant development. Still others produce compounds that make plants more resistant to drought or more attractive to pollinators. The plants aren't passive recipients, they're responding to fungal signals, sometimes encouraging fungal growth, sometimes suppressing it. The temporal dynamics of fungal chemistry add another layer of complexity. Fungi don't produce all their compounds all the time. They respond to circadian rhythms, seasonal changes, the presence of other organisms, nutrient availability, and countless other factors. A fungus might produce antibiotics only when competitors are present, toxins only when threatened, attractants only when ready to reproduce. This temporal regulation involves sophisticated genetic control. Fungi have epigenetic mechanisms, ways of turning genes on and off without changing DNA sequences that allow rapid responses to environmental changes. They can inherit these epigenetic changes, passing on not just genes but patterns of gene expression. A fungus exposed to a toxin might pass on resistance, not through genetic mutation but through epigenetic memory. The implications for biotechnology are profound. If we can understand and control fungal secondary metabolite production, we could have biological factories producing any compound we need. Instead of complex chemical synthesis, we could have fungi producing pharmaceuticals, industrial chemicals, even fuels. Some companies are already doing this, using engineered fungi to produce vanilla, leather-like materials, even meat substitutes. But there's so much we don't understand. Why do fungi produce such diverse secondary metabolites? How do they regulate this production? How do they avoid poisoning themselves with their own toxins? How do they coordinate chemical production in networks spanning acres? These questions touch on fundamental aspects of biology, chemistry, and ecology. The invisible chemistry of fungi shapes our world in ways we rarely appreciate. Every antibiotic that saves a life, every loaf of bread that rises, every forest that breathes, every piece of plastic that degrades, all involve fungal chemistry. The air we breathe carries fungal VOCs. The soil we walk on is transformed by fungal enzymes. Our own bodies host fungal chemists whose activities affect our health. As we face challenges like pollution, climate change, and resource depletion, fungal chemistry offers solutions. Fungi can clean our messes, produce our materials, even help regulate our climate. But to harness these capabilities, we need to understand fungi as chemists, not just what compounds they produce, but how and why they produce them. The invisible chemists are all around us, conducting experiments that have been running for hundreds of millions of years. They've solved problems we're just beginning to understand, created compounds we can't yet synthesize, developed systems we can barely imagine. In their invisible chemistry lies visible potential for medicine, for industry, for agriculture, for the environment. We just need to learn their language, understand their methods, and perhaps, with appropriate humility, become their students in the ancient art of biological chemistry. In the shadowed depths of forest floors and hidden beneath our feet lies one of Earth's most powerful climate regulators, a vast network of fungal threads that quietly orchestrates the planet's carbon cycle. These microscopic architects work tirelessly, weaving together ecosystems while performing an environmental service so profound that scientists are only beginning to grasp its full significance. The relationship between fungi and carbon storage represents one of nature's most sophisticated engineering marvels. Mycorrhizal networks, those intricate webs of fungal hyphae that connect plant roots across vast distances, function as massive carbon sequestration systems. Through their symbiotic partnerships with plants, these fungi receive sugars and other carbon compounds from their photosynthetic partners, effectively transforming atmospheric carbon dioxide into stable, soil-bound organic matter. The process begins when plants photosynthesize, capturing carbon dioxide from the atmosphere and converting it into sugars. A significant portion of these carbon-rich compounds, sometimes up to 30% of all the carbon a plant captures, travels down through root systems and into the waiting hyphal networks of mycorrhizal fungi. This carbon doesn't simply disappear. Instead, it becomes incorporated into fungal biomass and is eventually deposited into soil as organic matter, where it can remain sequestered for decades or even centuries. Consider the vast boreal forests of Canada and Russia, where ectomycorrhizal fungi form partnerships with countless conifers. These fungal networks extend far beyond individual tree root systems, creating underground carbon highways that span entire forest ecosystems. Recent research has revealed that a single fungal network can extend across thousands of square meters, with individual hyphal strands finer than spider silk yet stronger than steel cables, all working to transport and store carbon throughout the forest floor. The scale of this carbon storage is staggering. Scientists estimate that mycorrhizal fungi sequester approximately 5 billion tons of carbon annually, roughly equivalent to 13% of global fossil fuel emissions. Yet this figure likely represents a conservative estimate, as researchers continue to discover new mechanisms through which fungi capture and store atmospheric carbon. As global temperatures rise and weather patterns shift, fungal communities demonstrate remarkable adaptability, though not without consequences for the broader carbon cycle. Many fungal species possess an inherent flexibility that allows them to adjust their metabolic processes, reproductive strategies, and even their partnerships with plants in response to changing environmental conditions. In warmer climates, some fungi increase their activity rates, accelerating both carbon uptake and decomposition processes. This creates a complex dynamic where fungi simultaneously capture more carbon from their plant partners while also breaking down organic matter more rapidly. The net effect varies dramatically depending on the specific fungal communities, soil conditions, and ecosystem characteristics involved. Arctic regions provide particularly striking examples of fungal adaptation to climate change. As permafrost thaws and previously frozen soils become accessible, pioneer fungal species rapidly colonize these newly available habitats. These hardy organisms establish new networks in soils that have been locked in ice for millennia, beginning the process of carbon cycling in environments that were previously biologically inactive. However, shifting fungal communities also create uncertainty in global carbon calculations. As traditional fungal partnerships dissolve due to changing temperature and precipitation patterns, new associations form. Some of these novel partnerships prove more efficient at carbon storage, while others result in increased carbon release. The overall trajectory of these changes remains one of the most pressing questions in climate science. Drought conditions reveal another aspect of fungal climate response. Many mycorrhizal networks demonstrate remarkable resilience during water stress, maintaining carbon transport even when their plant partners struggle to photosynthesize. This persistence allows fungal networks to serve as carbon reservoirs during difficult periods, potentially buffering ecosystems against the worst effects of climate variability. The relationship between fungi and climate change operates through complex feedback mechanisms that can either accelerate or moderate global warming trends. Understanding these feedback loops has become crucial for predicting future climate scenarios and developing effective mitigation strategies. When soil temperatures increase, decomposer fungi accelerate their breakdown of organic matter, releasing stored carbon back into the atmosphere as carbon dioxide. This creates a positive feedback loop where warming leads to increased carbon emissions, which in turn contributes to further warming. In some northern ecosystems, this process has already begun to transform carbon sinks into carbon sources, fundamentally altering regional carbon balances. Conversely, mycorrhizal fungi can create negative feedback loops that help stabilize climate systems. As atmospheric carbon dioxide levels rise, many plants increase their photosynthetic activity, producing more sugars that they share with their fungal partners. These additional carbon inputs allow fungal networks to expand and deepen, potentially increasing long-term carbon storage capacity. The extent to which this negative feedback can offset positive feedback loops remains a critical area of ongoing research. Precipitation patterns add another layer of complexity to these feedback systems. Excessive moisture can lead to anaerobic conditions that favor different types of decomposer organisms, potentially altering the balance between carbon storage and release. Meanwhile, drought stress can cause both plants and fungi to reduce their carbon exchange, temporarily slowing both carbon sequestration and decomposition processes. The timing of these feedback responses varies dramatically across different ecosystems and fungal communities. Some changes occur within days or weeks, while others unfold over decades. This temporal complexity makes it challenging to predict how fungal carbon dynamics will respond to specific climate scenarios. Yet understanding these patterns remains essential for accurate climate modeling. Despite the uncertainties surrounding fungal responses to climate change, these remarkable organisms offer genuine hope for addressing global carbon challenges. Scientists and engineers are beginning to harness fungal capabilities for innovative climate solutions, ranging from enhanced soil carbon storage to novel carbon capture technologies. Researchers are developing techniques to optimize mycorrhizal partnerships in agricultural settings, potentially transforming farmland into significant carbon sinks while maintaining or improving crop productivity. By carefully selecting fungal strains and managing soil conditions to favor carbon storage, farmers could play a crucial role in climate mitigation while continuing to feed growing populations. Urban applications of fungal carbon storage show equally promising potential. Mycorrhizal networks in city parks, green roofs, and urban forests can sequester substantial amounts of carbon while providing additional benefits, such as improved air quality, stormwater management, and urban heat reduction. Some municipalities are beginning to incorporate fungal considerations into their climate action plans, recognizing the potential for biological carbon storage in urban environments. Biotechnology companies are exploring ways to engineer enhanced carbon storage capabilities into fungal systems. These approaches range from selecting naturally occurring high-carbon storage strains to developing synthetic biology applications that could dramatically increase fungal carbon sequestration rates. While such technologies remain in early development stages, they represent potentially powerful tools for large-scale climate intervention. Forest restoration projects increasingly incorporate mycorrhizal considerations, recognizing that successful reforestation depends heavily on re-establishing healthy fungal networks. By inoculating new plantings with appropriate fungal partners and managing soils to support fungal growth, restoration practitioners can create forests that not only sequester carbon in their biomass, but also build substantial below-ground carbon reserves through fungal activity. The path forward requires continued research into fungal ecology, climate interactions, and carbon dynamics. As scientists develop more sophisticated understanding of these underground networks, new opportunities for climate solutions will undoubtedly emerge. The fungi that compose our climate systems may well hold keys to stabilizing Earth's carbon balance, offering hope that grows quietly in the dark, beneath our feet, in the hidden realm where carbon finds its long-term home. In laboratories around the world, scientists observe behaviors that challenge our fundamental understanding of intelligence, memory, and decision-making. Organisms without brains navigate complex mazes, remember past experiences, make strategic choices about resource allocation, and coordinate activities across vast networks. These remarkable capabilities emerge not from neural circuits, but from the distributed intelligence of fungal networks, a form of cognition so alien to our experience that it forces us to reconsider what it means to think, remember, and perhaps even to be aware. The yellow blob of Physarum polycephalum spreads across the laboratory petri dish like spilled paint, its edges constantly shifting and pulsing. To the casual observer, this slime mold appears to be nothing more than animated protoplasm. Yet researchers have documented behaviors that would impress even the most sophisticated artificial intelligence systems. This single-celled organism can solve complex optimization problems, navigate intricate mazes, and design efficient transportation networks, all without possessing a single neuron. When researchers place food sources at opposite ends of a maze and introduce Physarum at the starting point, the organism initially explores multiple pathways simultaneously. Its searching tendrils probe dead ends, investigate false leads, and gradually map the entire maze structure. Over time, however, the slime mold withdraws from unsuccessful routes and reinforces the pathways that lead most efficiently to food sources. The final result is a network that connects all food sources through the shortest possible routes, effectively solving the maze through a process of distributed exploration and optimization. This problem-solving capability extends far beyond simple maze navigation. Japanese researchers demonstrated Physarum's network optimization abilities by recreating a map of the Tokyo metropolitan area using oat flakes to represent major cities. When the slime mold was allowed to grow and connect these food sources, it produced a network structure remarkably similar to the existing Tokyo rail system, a transportation network designed by teams of engineers over decades of planning and development. The mechanism underlying this intelligence operates through dynamic feedback loops within the organism's cytoplasm. As Physarum explores its environment, successful pathways become reinforced through increased cytoplasmic flow, while unsuccessful routes experience reduced flow and eventual abandonment. This creates a physical memory of successful strategies embedded within the organism's structure, allowing it to continuously optimize its network architecture in response to changing conditions. Other fungal species demonstrate equally impressive problem-solving abilities through different mechanisms. Mycorrhizal networks can assess soil conditions across vast areas, identifying optimal locations for resource extraction and allocation. These networks dynamically adjust their structure based on environmental feedback, growing new connections where resources are abundant while reducing investment in less productive areas. The concept of memory typically conjures images of neural networks storing information through synaptic connections, yet fungal organisms demonstrate sophisticated memory capabilities through entirely different mechanisms. Fungal networks can retain information about past experiences, learn from environmental changes, and modify their future behavior based on historical events, all through chemical modifications and structural adaptations that persist across time. Epigenetic mechanisms provide one of the primary means through which fungi store and recall information. When fungal networks encounter environmental stresses such as drought, temperature extremes, or pathogen attacks, they respond by modifying the expression of specific genes without altering the underlying DNA sequence. These epigenetic modifications can persist for extended periods, effectively creating a molecular memory of past environmental conditions. Research on Neurospora crassa, a bread mold commonly used in genetic studies, has revealed sophisticated memory systems that operate across multiple timescales. Short-term memories involve temporary changes in gene expression that last for hours or days, allowing the fungus to respond appropriately to immediate environmental conditions. Medium-term memories persist for weeks or months through more stable epigenetic modifications, while long-term memories may involve structural changes to chromatin that can last for years or even be passed on to subsequent generations. The practical implications of fungal memory become apparent when considering how these organisms respond to recurring environmental challenges. A fungal network that has previously experienced drought stress will often modify its growth patterns and resource allocation strategies in ways that better prepare it for future water shortages. This preparation occurs even when current conditions are favorable, suggesting that the network retains and acts upon information from past experiences. Mycorrhizal networks demonstrate particularly complex memory systems that operate across entire forest ecosystems. These networks can remember the locations of reliable plant partners, the seasonal patterns of resource availability, and the historical occurrence of disturbances such as fires or insect outbreaks. This ecological memory allows fungal networks to make informed decisions about resource allocation and network expansion based on decades of accumulated experience. The chemical basis of fungal memory involves sophisticated signaling cascades that translate environmental information into persistent molecular modifications. Stress-responsive proteins, regulatory RNAs, and chromatin-modifying enzymes work together to create stable memory traces that influence future behavior. This molecular memory system operates without centralized control, instead emerging from the collective activity of countless individual cells within the fungal network. Every moment of a fungal network's existence involves countless decisions: where to allocate resources, which partnerships to maintain, how to respond to threats, and when to reproduce. These decisions emerge from complex information processing systems that integrate environmental signals, assess risks and opportunities, and coordinate responses across vast networks of interconnected organisms. Resource allocation represents one of the most fundamental decision-making challenges facing fungal networks. With limited energy and nutrients available, these organisms must continuously choose how to distribute resources among competing demands: growth versus reproduction, exploration versus exploitation of known resources, investment in defensive compounds versus expansion into new territories. The decisions that emerge from these trade-offs determine not only the survival of individual networks, but also the structure and function of entire ecosystems. Mycorrhizal networks demonstrate sophisticated resource allocation strategies that would challenge even experienced economists. These networks can assess the relative value of different plant partners, adjusting their level of investment based on the quality and quantity of resources each partnership provides. When a plant partner becomes stressed or less productive, the fungal network may reduce its investment in that relationship while simultaneously increasing support for more reliable partners. Fungal networks possess extraordinary capabilities for risk assessment and decision-making. Before expanding into new territories, they send out exploratory hyphae to assess soil conditions, resources, and competition. These hyphae act as biological sensors, transmitting data back to the network through chemical signals. Rather than centralized control, decisions emerge through distributed consensus. Each hyphal segment reacts to local conditions while integrating broader network feedback. Fungi also exhibit temporal intelligence, balancing short-term survival with long-term goals. Their seasonal patterns of resource allocation reflect strategic, time-sensitive planning. These behaviors, along with memory formation and adaptive learning, challenge traditional views of intelligence and consciousness. If fungi can solve complex problems without a brain, might consciousness be more about integrated information than individual awareness? Some researchers now propose consciousness as a spectrum. Fungal networks may represent a distributed form of awareness, millions of local sensing events coordinating to create coherent, goal-directed behavior across space and time. These systems meet many criteria associated with conscious entities: information, integration, adaptive learning, and temporal continuity. Still, the question of what it feels like to be a fungal network remains deeply mysterious. If there is awareness, it is likely nothing like ours. Rather than a unified self, fungal consciousness may resemble a vast sensing field, alive with chemical gradients and environmental cues. Our relationship with fungi is far deeper than most realize. We walk on them, breathe their spores, and rely on their underground labor. Mycorrhizal fungi form symbiotic partnerships with plants, cycling nutrients, supporting root systems, and sustaining entire ecosystems. These threads are everywhere beneath forests, gardens, and city parks, silently holding life together. Fungi teach us that true success lies in patience, collaboration, and service to the whole. Rather than dominating, they grow slowly, adapt to hardship, and form resilient networks that have survived mass extinctions. Mycorrhizal fungi thrive by supporting their plant partners, showing us the value of mutual benefit and long-term thinking. For fungi, decay is not an end, but a vital transformation. As part of this ancient system, humans hold both responsibility and opportunity. Our actions impact these hidden networks, but by working with them, we can restore ecosystems, grow food sustainably, and reduce waste. Fungal wisdom offers more than practical lessons, it brings comfort. Beneath us, invisible threads connect all life. Every mushroom is a signal of this quiet intelligence, reminding us we are never truly alone. Even in darkness, life continues, patient, connected, and endlessly creating the conditions for growth. The ground beneath us is not dead, it is deeply alive,